Hey, Mr. P here. In this video, we're going to talk about the chemical requirements for growth, specifically microbial growth. Okay, in the last video, we talked about the physical requirements for growth, which were like osmotic pressure, pH, temperature, those kinds of environmental physical things. Now we're going to dive into what are some chemicals and molecular things, molecules that are required for microbes in order to grow effectively. Okay, so as was discussed in last video, we talked about the physical requirements, which can be seen on the left side of the screen, right? We talked about the different classes of bacteria and what temperature ranges, both kind of the minimum temperature range, but also kind of the maximum, and a little discussion about optimum temperature range. Obviously, there are a variety of microbes on the planet, and there are a variety of different environmental conditions that are specific to temperature. And we also talked about osmotic pressure, we talked about the pH and acidophiles, and now we're going to dive into, like I said, the right side of the screen and talk about what are some chemicals, specifically what are elements, and what elements need to come together to form compounds and molecules, and what molecules are required in order for microbes to grow effectively. And the first one we're going to talk about is this idea of carbon. So you can see that carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur are the big six compounds, or the big six elements that are required for all microbial metabolism in a variety of different capacities. So we'll talk about those. Now, carbon is obviously the first one up, and carbon is by far one of the most important requirements for microbial growth, because all of the microbial metabolism and all of the metabolic activities that happen both in prokaryotic cells but also eukaryotic cells is dependent on carbon. Now, carbon is the structural backbone of basically all organic compounds that make up all cellular components. And so if we're talking about DNA, it's carbon-based. If we're talking about proteins, they're carbon-based. If we're talking about glucose and different sugars for energy production, those are all carbon-based. Proteins are carbon-based. Okay, a lot of things that make up structural and functional materials within the cell are all based on carbon. And then we have terms like chemoheterotroph and autoheterotroph, okay, or autotroph. But chemoheterotrophs get most of their carbon from the source of their energy. Organic materials such as proteins, carbohydrates, and lipids. Okay, chemoheterotrophs, those are heterotrophs that must consume, and they get the energy that they produce in carbon form from what it is that they ate, organic material wise. Chemoautotrophs and photoautotrophs derive their carbon from carbon dioxide, and there is just a slight difference between chemoautotrophs and photoautotrophs based on what energy source they utilize in order to derive the carbon from carbon dioxide. Okay, you can see this picture. This is a typical cell. It shows you carbon in a variety of different capacities. You obviously can see sugars, which is an input. That would be like glucose or lactose or galactose. Those are all going to be carbon-based. Okay, in fact, these six little black dots are representing carbon. And so carbon goes in, CO2 can go in, CO2 can go out in different areas. ATP has carbon in it that is produced. These different 3-carbon products like pyruvate and glyceraldehyde 3-phosphate, G3P or PGA, all of those are going to have carbon in them as well. So there are a variety of different formats that carbon can kind of reside in, but all of them are important for microbial growth, and that's why we're talking about them. Okay, moving on to nitrogen, sulfur, and phosphorus. Those are other really important chemical requirements when we're talking about microbial growth. And in addition to carbon, we need to utilize nitrogen, sulfur, and phosphorus as elements to synthesize cellular materials, like, for instance, DNA, which is deoxyribonucleic acid. That is not only going to be carbon-based, which we already talked about, but it's also going to include nitrogen in the form of their nitrogenous bases. You can see those right here in the center of this ladder. Obviously, oxygen is a really big, important component to not only the backbone, but also in the bases. Phosphorus is a really important component to the backbone. You won't see phosphorus in the actual interior of the molecule. It is not part of the nitrogenous base, but it does make up a substantial portion of the backbone. And then you have hydrogen, which is going to be kind of flooded throughout. All of these are really important for the structural components of DNA. You will also see protein synthesis requires sulfur and phosphorus and nitrogen, oxygen, and carbon while we're at it. But these compounds over here are all of the 20 different amino acids that are naturally occurring. And the reason we're talking about amino acids when we talk about protein synthesis is that amino acids are the building blocks of protein. And so as we link these amino acids one by one by one by one, we're creating an amino acid chain. And that amino acid chain has to be folded based on some interactions between the nonpolar and polar, or the negative and positive charges of these amino acids, in order to provide structural and functional ability for that protein. Okay, but in both cases, DNA and protein synthesis require substantial or considerable amounts of nitrogen, phosphorus, and carbon. Okay, nitrogen, sulfur, phosphorus, carbon, really important in the production of organic compounds, which are the basis for life. Okay. Trace elements. Trace elements are a requirement for growth. You can see that these are different elements. You have basically copper and cobalt and iron and manganese and zinc and nickel. All of these are going to be really important when we are talking about the production of different metabolites. Okay. They're called trace elements because microbes require very small trace amounts of these mineral elements, but they are, while they need them in small amounts, essential for the correct functioning of specific enzymes. And so you'll see when you look at these different functions that all of them, or most of them, the overwhelming majority, ends in ase. Okay, all of these ase suffixes designate enzymes. Okay, so it doesn't matter what trace element you're talking about. If it is a function or functions as one of these ase names, it is an enzyme, and so it is an essential metabolite or an essential component to the success of that particular enzyme, usually as a cofactor, which means it kind of not only turns on or activates the enzyme, but it actually structures it in a way that allows it to be conducive to the function that that particular enzyme has. Okay, so we've talked through carbon, we've talked through amino acids, and a variety of different chemical components like carbon, nitrogen, phosphorus, and sulfur. Now we need to talk about oxygen requirements. Okay, oxygen obviously is an important chemical requirement, not only for eukaryotic cells, but we also have oxygen requirements for microbial growth. And those oxygen requirements are usually toxic, right? So there are a variety of different toxic forms of oxygen, and we're going to kind of talk through these. Don't assume that oxygen is always good for the living organism, okay? Because I think that there's a misconception where you hear oxygen and you automatically assume that it's a requirement and that it is considerably more important than a lot of other things. Like it's up there ranked with water, and it is, but there are a lot of toxic forms of oxygen that we really need to talk about. So we're accustomed to thinking of molecular oxygen, O2, as a necessity of life, but it is in a sense a poisonous gas, and we're going to talk why that is in a minute. Okay? Very little molecular oxygen existed in the atmosphere during most of Earth's history. In fact, it is possible that life could have or could not have arisen had oxygen been present. Because oxygen wasn't present in our early atmosphere, or if there was very little molecular oxygen, it makes sense that life adapted and overcame or was kind of naturally adapted to life without oxygen first. Okay, there's a lot of kind of evolutionary ideas or evolutionary evidence that suggests that a lot of the organisms, specifically microorganisms, adapted and evolved to not using oxygen first, and only once molecular oxygen became more abundant in the atmosphere did microbes kind of learn to tolerate and or exploit that as a particular chemical requirement. Okay? Many current forms of life have metabolic systems that require oxygen. Again, the key word is current. There are a lot of current forms of life, like current microbes and current aerobic organisms like us, that now require oxygen for aerobic respiration, but that wasn't always the case. Hydrogen atoms that have been stripped off from organic compounds, aka the food that you eat, are combined with oxygen that you breathe in to form water, which is why we expel water in the form of water vapor. Okay, we are actually stripping low energy electrons off of the food we eat, combining it with the oxygen that we breathe in order to form water vapor that we expel. Now, why do we expel water if water is such an important kind of chemical requirement? It's because we need to have a system in place that allows us to release low energy electrons so that we don't have low energy electron accumulation. This process yields a great deal of energy, which is in the form of ATP, while neutralizing a potentially toxic gas, which is oxygen. Okay, and so if you really kind of step back and understand kind of what is being said, that not only do we have a way of exploiting this energy form, which is electrons, from the food that we ingest, we simultaneously adapted and evolved to this toxic gas that was kind of increasing in abundance in our environment by giving or handing off those low energy electrons to the oxygen gas in order to not only rid our bodies of the low energy electrons, it also simultaneously rids our body of the potentially toxic gas, which is oxygen. So it's like killing two birds with one stone, so to speak. So what are the different forms of toxic oxygen? Well, we have a singlet oxygen, which is basically half of an O2. This is normal molecular oxygen that has been boosted into a higher energy state and is extremely reactive. Okay, it's just a single oxygen in the form of a singlet oxygen. We have superoxide radicals, which are formed in small amounts during the normal respiration of organisms that use oxygen as a final electron acceptor like us, that forms water. Many bacteria growing in the presence of these superoxide radicals must produce an enzyme called superoxide dismutase, or SOD, to neutralize them, which is really, really important. Okay, basically, if we step back and we talk about if a microbe utilizes oxygen as an aerobic respirer, uses oxygen as their final electron acceptor to hand those low energy electrons off, they will simultaneously produce this superoxide radical, which is a toxic chemical, right? It's forming toxicity, it's increasing toxicity in the cell. And so in order to combat that toxicity, they have to create this particular enzyme called superoxide dismutase to neutralize them. And so you can kind of see this particular equation, right? These superoxide radicals are basically neutralized to form hydrogen peroxide, which we'll talk about in a minute as well. The hydrogen peroxide produced contains the peroxide anion and is toxic and requires an enzyme called catalase in order to neutralize it, which converts it into water and oxygen, which is just your natural non-toxic oxygen. So once these superoxide radicals are broken down, it produces hydrogen peroxide, which includes the peroxide anion, which has to be broken down further using an enzyme called catalase. So we have a series of enzymes that have to break down these different toxic forms of oxygen in order to get to a form that is not only neutral but is able to be released or able to be reduced, right? Water obviously is very inert. It's not something that is going to be very reactive, right? It's going to be very beneficial for the cell. And so we have this SOD, or superoxide dismutase, which is going to remove the superoxide radicals, but simultaneously producing hydrogen peroxide. Then they use catalase to take this hydrogen peroxide and reduce it to water, which obviously is beneficial and is something the cell can use. The hydroxyl radical, which is an OH, is also kind of a toxic compound or a toxic form of oxygen. The hydroxyl radical is another intermediate form of oxygen and probably the most reactive. It is formed in the cellular cytoplasm by ionizing radiation. Most aerobic respiration produces trace hydroxyl radicals, but they are transient and can very easily diffuse out of the cell. That's really, really important. They don't really typically stick around. Okay, not going to belate or kind of talk about this a lot. Just I wanted you to be aware that there are toxic forms of oxygen, and it is important to kind of identify those and to at least be cognizant that they exist. Now, still sticking with the oxygen, obviously oxygen is a gas and it diffuses in and out of media very readily and very easily because it is small. But when we have these agar deeps, right, if we pour agar into a test tube and we seal them, or if we leave them open, oxygen in the air is actually going to be allowed to diffuse into them. And the longer you allow the oxygen to diffuse, the longer it will go. Okay, and so the areas on top obviously are going to have high oxygen, and the areas at the bottom are going to have low oxygen, and so it forms an oxygen concentration gradient. Now, if we talk about what the different classes of microbes are and why they grow in the areas they do, it requires a little bit of explanation. So, obligate aerobes. These are organisms that require O2. They are obligated to be aerobic, meaning they're obligated to have oxygen present. So growth occurs only where high concentrations of oxygen have diffused into the media, and that's important to note that they just require oxygen, and so they have presence of enzymes catalase and SOD. We just talked about what those are. Okay, catalase obviously going to break down the hydrogen peroxide. Superoxide dismutase is going to break down those superoxide radicals. And so they allow toxic forms of oxygen to be neutralized, and they therefore can use oxygen. So they are only going to grow in areas where oxygen is very high. Obligate anaerobes are the exact opposite. They are obligated to be anaerobic, meaning they are killed by oxygen. Growth occurs only where there is no oxygen, and as you can guess, they lack the enzymes of catalase and superoxide dismutase, and so if they lack those enzymes, they cannot neutralize them, and so those toxic forms of oxygen are toxic, right? They kill the bacteria, so they cannot grow where oxygen is present. Just think of it, oxygen is basically a poisonous gas for them. They cannot tolerate it, and they cannot neutralize it. Facultative anaerobes grow better with O2, which is why you see the majority of the growth like up in the top, but they also can kind of grow throughout, even in areas where oxygen is very sparse or where oxygen is completely not diffused. And so they grow better with O2, but they can live without it. So they can kind of tolerate no oxygen, but they obviously prefer it, and they live better in places where oxygen is present. Growth is best where most oxygen is present, but occurs throughout the tube, and they do have the presence of the enzymes catalase and SOD, so they can allow the toxic forms of oxygen to be neutralized, but they've kind of learned to grow in areas where oxygen isn't present. Okay? And then aerotolerant anaerobes, so these are anaerobic, but they are unaffected by O2 levels, so they just have kind of learned to tolerate oxygen. And they do have presence of one enzyme, SOD, but they don't have catalase, and so they can't completely neutralize the toxic forms of oxygen, but they can semi-neutralize it, and so they can kind of just grow wherever they want. Okay? Again, this is just a different way to kind of highlight where the growth is taking place, and it gives you kind of a snapshot on whether or not these particular groups have SOD or catalase. So obviously if there is SOD and catalase, then they can neutralize all toxic forms of oxygen and they grow where oxygen is present. If they have no SOD or no catalase, then they obviously cannot neutralize oxygen and they grow only in the absence of oxygen because oxygen kills them because it acts as a toxic. The only one we haven't talked about is this group because it wasn't included on the picture on the slide before. But there is a term or another class called microaerophiles, and those occur, or the growth only occurs where low concentrations of oxygen has diffused into the medium, and that's just because they semi or potentially sometimes have catalase but sometimes don't. And so if this is the agar, they actually grow in a specific band based on what specific range of oxygen is present. And so if they are growing towards the top, they're going to be a little more aerotolerant, but if they grow, like if the band's down here, they're going to be a little less aerotolerant. But these are microaerophiles. They are growing kind of in the middle, but they require a specific range of oxygen, and so they just grow in a particular band based on wherever the diffused oxygen concentration that they grow best is. Okay, produce lethal amounts of toxic forms of oxygen if exposed to normal atmospheric oxygen, which is why you do not see them grow up here, but they can grow where oxygen is a little less abundant. Okay, that's all. If you have questions, bring them to class. Otherwise, see ya.